So we've gone over the parts of the cell. We've gone over the cell membrane. Now we've got to figure out how the cell actually runs, how we can have all these reactions happen in it. Because really all a cell is a bunch of chemicals that got together and started thinking they were alive. So I know you guys are really excited about this. Back to chemistry. I know, it's biology. Why do we have to keep going back to chemistry? It's because all of this stuff is based on basic biological precepts. And you can tell it's chemistry because you have flasks with different colored liquids. Hey, right. The big thing you'll start noticing now is I just keep repeating myself. For instance, you have seen this exact same slide before. I believe we've talked about it at the end of chemistry. We talked about how briefly, very, very briefly, all chemical reactions are going to require energy. We talked about most chemical reactions require a catalyst, and how chemical reactions will keep moving in a certain direction until they reach equilibrium, and that most of these reactions are going to happen dissolved in water. And that's where I really emphasized the water aspect, and I kind of glossed over the rest. Like, you weren't even tested on those things. That's what's coming up on this exam. This test. We're going to look at how energy goes into a system, what a catalyst is, and what chemical equilibrium is. Basic vocabulary I need you to understand is that on the left side of the equation, you have reactants. On the right side, you have products. Reactants can either combine or break apart to become products. I say that because I'm going to use those terms a lot today, and I need you to understand what I'm saying. If you don't understand a term I'm talking about, raise your hand and stop me so that I can explain it. Yes? The products and reactants part? What? Okay. Thanks, guys. No, seriously, that's good. That's really good. Reactants are what you're putting into a reaction. You know how you guys you've at some point in your life had some sort of chemical reaction happen by accident, right? Right, baking soda and vinegar, right? Yeah. All right, you take baking soda and vinegar. Those would be your reactants. You're putting them together. And the product's what comes afterwards. Does that make sense? Sorry, thanks for helping me explain that. No, and that's why I do ask you guys to stop me, because if you don't, there's going to be 10 other people that have no idea what I'm talking about. And I will happily stop and explain it. So we have reactants come together to form products, or reactants can be split apart to form products. So equilibrium. All reactions are going to move toward a state of equilibrium. They all want to equal a state where the rate of formation of products equals the rate of formation of reactants. That is, it's moving equally in both directions at the same time. It's just like an isotonic solution where the water is moving in both directions at the same time. A reaction will continue to proceed forward in one direction until eventually you hit a point where the amount of products being created equals the rate of formation of the reactants. So everything balances out. We talked about equilibrium when we talked about buffers too. So the rate of formation of hydrogen ions equals the rate of the destruction of hydrogen ions, or the binding of hydrogen ions. That's what allows the buffer to work. All reactions are trying to head toward this very, very stable state. So now we've got what's called our metabolism. Now we've talked about metabolism before. This is kind of cool, I think, because all we're doing is going over some topics we've already covered. We're just looking at them in slightly new ways. Like metabolism. Metabolism is the sum of all of the reactions within the cell, whether they're catabolic or anabolic. What does it mean to be catabolic? It's bad. You're thinking ions, cations and anions. Catabolism and anabolism. It does, actually, yeah. hold on. Metabolism is? Never mind. Okay, and metabolism is how you process things? Yeah, it is. It's all of the reactions in the cell. So you have two big types of reactions. Catabolic reactions and anabolic reactions. It's okay. You got it. You got this. No, don't worry about the pen. Yes.
Metabolism is breaking down a molecule.
Yep.
Anabolism is building.
Sorry, she got it first.
Sorry, I beat you.
Catabolism is breaking it down. Anabolism is building it back up. So when you have all of your catabolic reactions and all of your anabolic reactions combined, you have your metabolism.
Cats break it down and builds it up.
Yes. Have you ever heard of anabolic steroids? No?
But steroids build.
Right. Anabolic steroids build muscle. Let's see. That's right. Cats destroy things. Ooh. I have a stupid fat cat.
I love that cat.
Yeah, well, my stupid fat. I just bought an entire new living room set. You see where this is going, don't you? One of them.
Just return it.
No, I can't return it. I tried. Oh, are you talking about the cat or the?
Couch.
I tried the cat too. So right, there you go. That's a great way of remembering. Stupid cats freaking destroy furniture. Thus, catabolic reactions destroy things. Thanks. I'm keeping that one forever and ever. Anabolic builds things. So anabolic reactions build them up. So I don't know how you can remember that.
I'm going to remember the steroids right now.
Yeah, anabolic steroids. So, okay, how about this? A little bit further in. We're going to hit something you've already talked about and you should know backwards and forwards. And I'm going to totally call on Sarah for this. Yeah, you got this, Sarah. No, you got this, Sarah. Don't worry, you can always rely on Reagan. Why? Because you're the good one. I don't know. It's the best thing I could come up with on the spot. Oh. So, here we go, Sarah. Are you ready? What kind of reaction is going to take two molecules and put them together? A condensation reaction, right? So, I know. Okay, how about this? What kind of reaction will split things apart using water? Shh. What? Yeah. Sarah's got this. Hello. Hydrolysis. Right. A hydrolysis reaction. Okay, so we've got condensation reactions are building. So what kind of reaction are they? Anabolic or catabolic? Anabolic. Hydrolysis reactions are breaking them down. They're catabolic. Now get this, and we talked about this a little bit before, I think. What happens when you break bonds? What's released? Energy is released, right? And then when you're building something up, what happens with energy? It's stored. That's like the whole lecture today. Yeah. So, well, no, I'm going to give you some more terms, and we're going to see some graphs. But if you understand that concept of you break something, you release energy, you build something, you store energy, you got it. Then we're going to talk about ways of reducing the amount of energy it takes to build something. I'm so proud of you guys. So, we talked about this macromolecule called proteins. One type of protein, one category of protein are called enzymes. Enzymes speed up reactions in the cell. That's their whole job. They speed them up. So, Venn diagram. My daughter's been working a lot with Venn diagrams lately. So, they're on my mind. So we got large category of macromolecules. One subgroup of that are proteins. Sort of like that. And then overlapping the proteins would be the catalyst group. And then where catalysts and proteins overlap, you have enzymes. So enzymes are catalysts. They are also proteins that speed up reactions in the cell. Okay, so I used a lot of words there. Do you have questions? So if it speeds up a process, it's called a catalyst. An enzyme is a type of catalyst. Yes? Yeah, macromolecules. Because remember, there are 4 classes of macromolecules: proteins, carbohydrates, lipids, nucleic acids. There we go. So in order to understand how we can speed up reactions, we need to understand how energy works. So now we're moving even away from chemistry and talking a little bit about physics. But this is like baby level, not baby physics, but it's really at most middle school level physics. You don't need to know much physics for biology. We have to understand a little bit about energy. Energy is the ability to promote change and do work. And there are 2 broad types of energy: kinetic energy and potential energy. Has anybody never heard of these before? Okay, these are things that you've probably talked about. These are really easy to test on in SOLs. So the idea is, think of kinetic energy as the energy of motion, the energy of movement, and potential energy is that stored energy. So if looking at this, what is this thing called? A roller coaster. You have it building energy, storing energy, the potential energy increases, kinetic energy increases, potential, kinetic. And you'll notice there's a relationship between them. As potential energy decreases, kinetic energy increases, and vice versa. You can convert from one form of energy to another. That conversion, or the study of that conversion, is known as thermodynamics. You've probably heard of thermodynamics before. It's a really big, scary word. Thermodynamics. Yeah, thermo, so you're looking at heat, and dynamics, so the changing of heat. You don't need to know much about thermodynamics except for 2 things. The 2 laws, that's right. And that is the most important rule we have to remember. The first law of thermodynamics: energy can neither be created nor destroyed. This is really, really important. We all knew that, right? Energy is neither created nor destroyed. It's only transferred from one form to another in the universe. We don't know what happens in black holes. I'm not terribly worried about them. They're not going to affect the way biology works on Earth for a long time. Well, that's going to be a long time. And if it kills us, that's that. Don't worry about it. I was wrong. I'll admit it then. So what I want to go over as far as first law of thermodynamics is basic photosynthesis. Very, very basic. The sun provides light. We good on that? Everybody understood that? That light is coming at us in the form of waves or energy, kinetic energy. So kinetic energy is coming from the sun and striking the pigments in the chloroplast. These are called the chlorophyll. So you're taking energy, kinetic energy, it's striking a pigment, and that pigment converts that kinetic energy into potential energy of a bond. So we're building sugar here. We're using that kinetic energy to build sugar. So far, so good? Kinetic to potential. Now we're going to, what kind of reaction is that for building this molecule? Anabolic. It's building a molecule. Now we're going to take that sugar, and it gets transported over to the mitochondrion. What does the mitochondrion do? It's the site of aerobic cellular respiration, which means it's going to be take that sugar, using some oxygen, and break it. All right, so we've got potential energy in the bonds of glucose now being released as kinetic energy. So we're going from potential energy, kinetic energy of the sun to potential energy of the bonds of glucose to releasing that energy in the mitochondrion. To make ATP, you bind ATP together, you're using the energy from the glucose to build new molecules. So it's back to potential energy. We are not losing energy in the system. We are simply transforming it from one form to another. The second law of thermodynamics says that transferring or transforming energy will increase the amount of entropy, or the degree of disorder, the amount of unusable energy. So we're not losing energy. What we are losing is usable energy. It might be released as excess heat. It might be released as any other form of energy, but it is still there. It's just we can't use it. As far as entropy, it's increasing disorder in a system. There have been points in your life where you've been left home all by yourself, right? Yes. I know. It's wonderful. Have you noticed that no matter how much you try, you still end up having to clean the place at the end? It's because entropy always increases unless you put energy into the system. So you need to add energy to keep it clean. Otherwise, it's just going to fall apart all on its own. This is coffee art. I know, weird, isn't it? People take the time to do art in their coffee. Now, is that going to stick around for a long time? If your barista did this, one, give them a tip. In fact, just do that in general. They need it. But two, how long is that going to stick around? Yeah, like a minute, 2 minutes. It's even worse if you want to stir your coffee, right? You just walk. Yeah, you walk it from. You're like, oh, this is great. I have to take a picture of it right now because it's not going to last. Is it possible to make it last? No. It is. What? No. You got a spoon here. You could keep adding energy, stick the spoon in, and keep rebuilding it. Oh, no, stop combining together, just reworking it. But that takes a lot of energy. You're investing energy in the system. That energy that you're investing would prevent the entropy from overwhelming the system. So when you transform energy from one form to another, from kinetic to potential or back again, each time you do that, you're going to increase the degree of disorder, or you're going to lose a little bit of that energy in the process to the system. It still exists. It's still there. But you cannot use it. It is no longer usable. Now there's, yes?
So entropy is energy?
Energy is the degree of disorder in a system. Entropy is, how messed up things get when you're not taking care of it. You can prevent entropy by adding energy. Trying to raise a kid. If you don't invest energy, that kid's going to be messed up. Some of you guys are products of that. You totally understand. You're like, I was like, yeah, okay, I did kind of raise myself. The amount of energy, and there's been a ton of studies, the amount of energy you put into kids really gives them a positive, a good push in life. Like, you push too much, though, they become rebels. So, and then it comes down to who cares? This is all physics and chemistry, and this is biology. Stop talking about this stuff. The reason we need to care about this is because thermodynamics, these two laws, place constraints on what a cell can do. A cell can't violate physical laws. So we've talked about energy. Energy in the system is known as free energy. This is energy that you are able to use. Sometimes through entropy, you lose some of that energy as unusable energy, energy you can't use to do work. So free energy, what you use to do work, plus that unusable energy equals the total amount of energy in the system. That total amount of energy doesn't change. That's what remains constant, because energy is neither created nor destroyed. What changes is the amount of free energy and the amount of unusable energy. No, I won't have you do mathematical precepts asking you to take, here's this amount of energy, here's that amount of energy, calculate it. I don't think that's important for introductory courses. It is kind of important. It's just understanding the differences between the two broad types of chemical reactions. If a reaction doesn't need energy to begin, that is, if an action in its process releases some energy, it's called exergonic. Back to our earlier stuff. What kind of reaction could be described as exergonic? It releases energy. Catabolic reactions are going to release energy. And give an example of a catabolic reaction. An explosion, but I'm thinking, give an example of one we've talked about, a chemical description of one. I wish Sarah was here. It's the other one. It's the other one. Hydrolysis. That's okay when I say Sarah. Now everyone thinks condensation. I've trained you guys like Pavlov trained his dog. I'm like, Sarah, and you're like, condensation. No, it's hydrolysis. A hydrolysis reaction could release some energy. So condensation would be an endergonic reaction, otherwise known as what? It requires energy, so it's going to be anabolic. All right, exergonic reactions. Let me go over this before we get ahead. We've got the course of the reaction, so the reaction occurring over the time. Over here we have the reactants, those things going into the reaction. They start off with a lot of energy. The products at the end have less energy associated with them. The difference between it is the amount of energy that's released or given off. So you have a high state of energy, and then it ends at a low state of energy. So that energy was released to the system in an exergonic reaction. An example of an exergonic reaction might be, do you guys know what hot hands are? Yeah. Yes, you guys know what I'm talking about? Yeah, you open up this pack, and there's a reaction that begins to occur. Once it interacts with oxygen, suddenly they start to heat up, these little pads. And as they heat up, good, okay. As they heat up, they're giving off energy. That's an exergonic reaction. They're giving that energy to you. The flip side are endergonic reactions. These are going to require energy. They need energy to happen. They're also called anabolic reactions. So here you have reactants with a low amount of free energy. And in order to get them to come together, to get something new, you have to add energy to the system. The difference between the amount of energy in the reactants and the products is the amount of energy you need to start the reaction, to get it going. Okay, so now give me an example of an endergonic reaction. We've already done it. What? Yeah, it's an anabolic reaction. Okay, wait. Hey, Sarah, what is an example of an anabolic reaction? It's the same thing I always ask you. It is condensation. That is the answer. Whenever I call on you from here on out, the answer is condensation reactions. Okay. Okay, I'll call on somebody else for that. Colin Reagan? No, Reagan knows these things. She's the smart one. Endergonic reactions. So all of the combined endergonic and exergonic reactions put together will give you your metabolism. And they should yield a, hopefully, net zero energy total. Now, we constantly have to take energy into our system, though, right? How do we get energy? Not trying to trick you. Food. You eat things. You eat things, you break them down, you steal their electrons, and you make those electrons do some work for you. So, we've got to get free energy. We need energy to get these reactions going. We can get it from food, and we have to take that food and turn it into a usable cellular energy source. And that usable cellular energy source is adenosine triphosphate, or ATP. This is the major energy carrier in the cell. You can always recognize ATP, well, for the most part, because that TP, triphosphate, what does tri mean? 3. 1, 2, 3 phosphates. So get this. You're going to take that ATP and you're going to break it down. What kind of reaction will you use to break down ATP, to split it? A catabolic reaction. And chemically, what kind? If you're going to use water to do it. Hydrolysis. So for the hydrolysis of ATP, you're going to take a molecule of ATP and you're going to split it. You're going to break a bond. You're going to remove a phosphate. That's a catabolic reaction. It's going to give off energy. If it gives off energy, how would you describe it? Exergonic, catabolic, exergonic, hydrolysis. Any of these will be fine. So this ATP here gets split using water. Right. So the phosphate is cut off. The hydrolysis of ATP is how the cell gets a lot of its cellular energy. This is what moves energy around the cell. These are like little cellular batteries. They can move from one place to another. Just like you can charge a battery at a wall socket and then move it to like a remote control and then move it from there to something else. So if you can attach an endergonic reaction to an exergonic reaction, so you attach these reactions where you need energy to one that gives off energy, you can have spontaneous reactions. Let me break that down. There are endergonic reactions that need energy. You need to build something, right? So you need energy to do that. The energy you can use to build things comes from you breaking other things down. So you get energy by breaking something down, and that energy can then be transferred to another reaction. So you're moving energy from one form to another, from kinetic energy to potential to kinetic to potential. Each time you do that, you lose a little bit of the energy to the system, entropy. But you can do it. Energy is neither created nor destroyed. It's just lost. It's sort of just hanging out. It's just we can't use it. It might be released as heat or light, but we can't use it for running our reactions. That's why you have to, even though energy is neither created nor destroyed, you have to keep eating. You need to keep replenishing the usable energy. So you pair the hydrolysis of ATP with your other reactions. Questions so far? So we're going to start talking about enzymes, but before I talk about enzymes, I really want to get into, again, that idea of moving energy around. I do think of ATP as a cellular battery. You can recharge a cellular battery. That would be like building the ATP. How many of you guys use rechargeable batteries at home? Yeah. I think they're, in the end, they're way cheaper than having to keep buying new batteries. All right. They explode. It's not like a galaxy. You take these batteries out of the wall, you plug them into your remote control, your remote control starts working. You're using some of that energy. That energy is then decreasing in it. You can then, if you wanted to, move the battery from the remote, and you guys know you've done this at some point, to something else because, man, you need that other battery. So you can keep moving these batteries from one point to another, but each time you move the battery, you're losing a little bit of energy until in the end, you're out of energy. So you have to recharge it. Energy can move around a system. And truthfully, studying the way energy moves through a biological system, a whole system, is ecology. I know a lot of you guys remember studying ecology when you were in middle school and you learned about biomes and the tundra and how things work like that? It's way less interesting at higher levels. It's all about studying the way electrons move. Okay, so I want to talk about, I guess for the remainder of the lecture, enzymes. Enzymes are proteins, which means what are they made of? They're made by ribosomes. What are they made of? Amino acids. You did. Well done. You win. Enzymes, like we said earlier, are catalysts. A catalyst is something that speeds up a reaction without itself being altered or consumed in the process. As you can tell, I was up late trying to put the slide together. because that is not a biological catalyst. Google Images did not serve me well this time. What that is a catalytic converter on a car. But then I was like, I should remove this and really spend another half hour trying to find a picture of a catalyst. And I was like, no, this kind of works. Do you guys know what a catalytic converter does? No. Yeah, just magic. Yeah, it goes out and then the car doesn't charge the battery. Because you see arrows. So it breaks down pollutants. That's one of the things it does. Exhaust goes in. Carbon dioxide. So you've got hydrogen, carbon monoxide, and nitrogen oxide go in. Carbon dioxide and nitrogen go out. And that's where you get your greenhouse gas of carbon dioxide. But truthfully, it's a lot better than carbon monoxide. That could kill you. Right. This is a good trade-off.
But what about when you're... Oh, wait, never mind.
So what ends up happening is these molecules are broken down at a higher rate into those molecules in the catalytic converter than outside. But on a macro scale, let's not even talk about catalytic converters. Let's just talk about mechanics and car problems, because we've all had that. A catalyst is something that speeds up reaction without itself being altered or consumed. How many of you guys have gone driving and that little check engine soon light pops up? Yeah, pretty much all of us, because we all have really crappy cars. You like your car? You've got...
I just got a new car.
Oh, is it like brand new?
It's 17.
Seven? That's really new.
Yeah.
Did you get a check engine light on that?
It went away though. It fixed itself.
That's the question. What do you guys do when you get a check engine soon... Guys, what do you do when you get a check engine soon light up?
You freak out.
First thing you do, you freak out. You
And then you ignore it.
That's right. That's what we all do. Every single one of us will look at that thing and say, if I hit a pothole just right, it'll fix itself. Right? There is hope beyond hope that could occur. Or you assume the light itself is broken. Because it's an expensive fix, isn't it? So you drive along 24/7. This light stays on. Nothing gets fixed. Sometimes it might because you tighten your gas cap. But the idea is, so you tighten your gas. So there you go. So this light stays on until you take it to a mechanic or you fix the problem on your own. If you didn't tighten the gas cap, if you didn't take it to the mechanic, we're pretty sure it'd stay on in perpetuity. Now, how could, is it possible it would fix itself? Yes, it is possible. It is in the realm of possibility that your car could sit in a field and the raindrop could hit it just right and there's a vibration or something and the whole car gets fixed. That is absolutely possible. It's possible. It could take 1 billion years, but it is possible. The thing is, you can make it happen faster. How do you fix your car fastest? You get it fixed. You take it to the mechanic. What does the mechanic do? They open it up. They run a diagnostic. It's going to cost you money. Don't get me wrong. But they can make this car work. In the process, does it happen faster than it would have happened on its own? Yeah. Is the mechanic then altered or consumed in the process? That is, do they lose like a hand to your car? Not if they're a good mechanic. They're not consumed by it. It's not like your car eats the mechanic in order to get better. So the mechanic acts like a catalyst. They speed up the reaction. And then the cool part is they don't just fix your car and they're done. They can go on to the next car and the next car. So if you have one mechanic in a group, that one mechanic can fix multiple cars, making all these people's lives easier, happening much faster. A catalyst, like the mechanic, will speed up a reaction without themselves being altered or consumed. And this is where I like to say there's an intersection of politics and science, because you've probably heard the term catalyst outside of this class. Lots of organizations say they are a catalyst for change. In fact, I think John Tyler says that. We're like, we are a catalyst for change in the community. What people think catalyst means in modern parlance is they speed up change. But what they forget about is the second part. I went to a VCCS conference, Virginia Community College System conference, And one of the high muckety-muck vice chancellor, arch-chancellor, whatever they are, the people who are in charge, they're giving this speech. And they're talking about how the community college system is a catalyst for change in the region. And how we are all catalysts. And so I'm the jerk that just stands up and I'm like, I have a question. And at first they weren't going to call on me, so I started jumping and waving my hands around until they're like, oh, did you have something to say? And I was like, yes. Do you know what catalyst means? And they said, yes, it means we change things. Like, that's not all a catalyst means. A catalyst means we change, a catalyst will change something without itself being altered or consumed. So if we, at the college, are saying we are catalysts for change, what does that mean?
They're not changing, but they're changing other things.
Right. So that's like me saying, I'm a catalyst for change. I'm changing you, but I'm myself not going to change at all. That's not a catalyst. That's not a catalyst at all. It could be. Well, then I was thinking, you guys have had professors at some point in your life, I assume, who sort of represent this whole, I'm going to change you, but I'm not going to change. They have like those like yellowing notes that they read from. You've been in those classes, right? Yeah. So maybe we are. That is the way it works. The truth is, though, in a college setting, my interactions with you guys change me just as much as my interactions with you change you. I get you to learn things, but I'm learning all sorts of stuff, like what works and what doesn't, and what I have to do to help you on an individual level succeed. So I don't think we can say colleges and professors are catalysts for change because we are changing. So there we go. That's politics. So enzymes are catalysts. That means that enzymes are going to speed up a reaction without themselves being altered or consumed in the process. They increase the speed at which reactions occur by many, many times. At this point, I want to discuss the parts of an enzyme, because Reagan left and it's going to be easy to talk about when she's not here. So, there's the enzyme itself. The enzyme has distinct structures on it. It's got a shape. The shape of the enzyme is called its conformation. You need to know that the shape's called a conformation. It's kind of easy to remember because it's got the word form in it. So the form is the conformation. It's enzyme shape, shape of the enzyme, whatever you want to say. That's the shape of this particular one. They have lots of different shapes because each of them has a distinct form, giving them each a distinct function. Now, with this enzyme, you have a place where something can bind. That place where something can bind is called the active site. And then the thing that binds to the active site is called a substrate. So the enzyme has a specific shape, a conformation. Part of its conformation is an active site, and that active site can bind to a solute in the solution called a substrate. And it fits pretty darn well. Oftentimes what it'll do, like here we've got the enzyme, it's got an active site. Sucrose, a disaccharide, can bind to the active site. And that sucrose is split or broken apart into fructose and glucose. So the substrate is released, and you go back. You'll notice the enzyme itself doesn't change. It speeds up the reaction without itself being altered or destroyed. Say that again? We talked about receptors when we talked about the membrane. In the peroxisome, we have, what happens in the peroxisome? Do you guys remember? Speeds up things, breaks things down. What does it break down? Yeah, because it's on the slide. Breaks down hydrogen peroxide. It breaks it down 10 quadrillion times faster than it would normally. So one little bit of enzyme, little bit of this enzyme called catalase, breaks down hydrogen peroxide 10 quadrillion times faster. That number is almost unimaginably huge, which means that we don't have the effects of that free radical because it breaks down faster than it's being made. This catalase, this sort of enzyme shape here. What is catalase? It's a protein that breaks down hydrogen peroxide. It literally grabs hydrogen peroxide and rips it apart. Well, that's exactly what we have in our wounds. That's why so you take hydrogen peroxide, you put it in your wound, that thing goes along and rips the hydrogen peroxide apart. Yeah, so we are totally fine. Our wounds can handle hydrogen peroxide, no issue. It's bacteria that can't handle them because they lack this. Yep. Yeah, they wipe out things that don't have catalase. Here's why we need enzymes. When two atoms or molecules bond, their electron clouds repel each other. So the deal is, when I told you guys about covalent bonding, it should have made absolutely no sense. For some of you guys, it actually did make absolutely no sense. But the idea is you have one atom and another atom. You bring them together. And what I told you was, when you bring these two electron clouds together, they share electrons, right? And you guys are all like, yeah, that totally makes sense. That makes no sense whatsoever. Because if you have a negatively charged electron cloud and a negatively charged electron cloud, and you bring them together, what should happen? They should repel each other. But they don't. We're just like, but they bind covalently. The reason you need energy for reactions is because you need to overcome that repulsive force. If you were to take two magnets charged the same way and bring them together, they push each other apart, right? Can you get those magnets to squish together? Yeah, because you're strong enough to push them. Does that make sense? You add that energy to bring them together. And when you do that, then they stick. You have you were adding activation energy. Here, you're adding activation energy. The energy you need to overcome the repulsive force of those electron clouds. Oh, every single semester I do this. I meant to bring eggs. This would have been so cool if I had eggs. I'll go into what I'll do. Okay, so imagine, if you will, I had 2 eggs. This would have been brilliant. I would have moved this chair so you could see too. Okay, so I take 2 eggs and I put them on this desk, and they are not hard-boiled or any tricks like that. They're just straight up eggs out of the refrigerator. And I gently roll them toward each other. What will happen? They'll roll toward each other, they'll hit each other, and then bounce off. Just like if they had electron clouds. They have that repulsive force. They're going to bounce off of each other. How would I get them to stay together? That's right. You take them and you go swoosh. And then I would have been like, there would have been a splash zone for it. It would have been great. But the eggs then, once you get them together, do they come apart? No. They stay together. You added that energy, and that forces them to stay together. You need to add that activation energy to get these 2 atoms to stay together, to overcome the force of those electron clouds. Once you overcome that force, they stay stuck. You have a covalent bond. So the point of enzymes is to overcome that repulsive force. You have to add it. What enzymes do is they lower the amount of energy you need to overcome that bond. They lower the amount of activation energy you need. They make it easier. So enzymes decrease the activation energy necessary for a reaction to occur. Which is what this slide says. So here we've got ATP and glucose. The ATP and the glucose are going to bind to a substrate, to an enzyme at the active site. And then they're going to be brought together. A phosphate is transferred to the glucose, and ADP, adenosine diphosphate, is released. This speeds up this reaction. And the way this one speeds up the reaction is it literally will bring these 2 molecules together and then start straining the bond between the phosphate at the same time squishing it to the glucose. so that it makes the phosphate switch from one molecule to the next. You're just transferring a functional group. Pardon? No, it's cool. What? Any? Oh, the one that says, why do we need enzymes? No problem. Slide 26. So enzymes are going to be proteins that are made from amino acids that have that very, very specific shape, and that shape is called its conformation. This P shape here is its conformation, its size, its shape. The conformation determines exactly what that enzyme will bind to. It's very, very specific. They're not general. So on the enzyme, you have a place called the active site. The active site is where you're going to bind to the substrate, the thing that you're interacting with. Again, the form determines the function. So the form of that active site determines what substrate it can bind to. Now, that specificity of the form connecting to the specific enzyme is called the lock and key hypothesis. Enzymes are going to typically bind to one type of substrate. So the specific enzyme will bind to a very specific substrate, usually. And by controlling how much enzyme is produced and how much enzyme is removed from a system, you can control how efficiently the different metabolic pathways in your body work. So this is, again, lock and key. The substrates are very specific to the enzyme. Once a substrate binds to an enzyme, you have what's known as induced fit. The enzyme changes shape a little bit. Now, it's going to release. You're not going to permanently change the shape. But can I use this cup real quick? So the idea is, generally, I need this sort of C shape to fit on that cup, right? If I was to come on and put a C shape here, though, it's not going to be able to pick it up. What needs to happen is it needs to curl around a little bit more, tighten up a little. So in order to lift it, you have the C shape binds, and then it tightens, and then you can lift. So that's called induced fit. You're changing the shape. And the reason this happens is because, remember, what's this protein made of? Amino acids. And each amino acid has specific chemical properties associated with it. So those amino acids are going to interact with these substrates, causing them to shift around a little. You might have new disulfide bridges form. You may have new hydrogen bonds form, a new hydrophobic effect, or new van der Waals forces. So it changes the shape of the enzyme just a little bit, causing it to wrap a little bit tighter around the substrate. All right, so that's what enzymes are. Environmental factors will play a huge effect in how well these enzymes work, how efficient they are. And there are 4 big factors that affect enzyme function, 3 of which we're going to cover in lab. Enzyme concentration, the presence of inhibitor, temperature, and pH. So concentration, inhibitors, temperature, and pH are 4 factors that you're going to need to absolutely know. And you know there's a list here. It's going to be on the exam. I'm just saying, that's an easy question. So first off, I'm going to go through each of these. Enzyme concentration. If you have one enzyme in a solution, it speeds up the reaction, right? What happens if you have 2 enzymes?
Double speeds it.
That's right. It makes it go even faster. No, that's exactly right. That's what I was getting at. So if you have a single enzyme in solution, it speeds up the reaction. If you have more than one, if you increase the amount of them, increase the concentration, suddenly the reaction goes even faster. It breaks things down much more quickly. Does that make sense? On the other side, you might have inhibitors. Inhibitors slow down reactions. You have two broad types of inhibitors: competitive inhibitors and non-competitive inhibitors. A competitive inhibitor is a substrate that's in the solution that will bind to the active site. So if where did I just put that marker? There it is. Here's our normal substrate for our enzyme. If I was to have another inhibitor, it's got the same shape. If it was to bind there, it would stop the substrate from binding. You guys have heard of formaldehyde? Yeah. Formaldehyde prevents enzymes from working, which stops organisms from breaking down. So a competitive inhibitor competes with the substrate for the active site. So if you have a lot of competitive inhibitors present, the reaction rate decreases. A non-competitive inhibitor does not compete for the active site. It's non-competitive. It binds to what's known as the allosteric site, a different site. This is outside of the active site, and it causes a change in the shape of the enzyme. So if you had an allosteric site over here on the enzyme, and something comes in and binds there, when it binds, it will change the shape of the active site so it can no longer fit to the substrate.
So the substrate still doesn't bind.
Substrate still doesn't bind. Net result, no binding of the substrate, which stops the reaction. So the more inhibitors you have, the lower the reaction rate. Competitive inhibitors compete for the active site, stopping substrates from binding. Non-competitive inhibitors bind outside of the active site, but they still stop substrates from binding. So the more inhibitors you have, the lower the reaction rate. You have a whole bunch of other stuff going on in the environment, though. You might have pH. What's pH a measure of? The amount of hydrogen ions present. And you might have temperature. Do you guys know what temperature is a measure of? The measure of movement, the measure of kinetic energy. So you might have these two conditions, pH and temperature, that will impact the reaction rates dramatically. First of all, temperature. Oh, by the way, sorry, last slide. There's this word called optimal. Optimal means the best. There are like the best possible conditions. So enzymes work best in the environment in which they've evolved. What is your body temperature? Roughly. Fahrenheit, I don't care. 98.6. 96.1 if you're running cold, that's cool. People have different temperatures, surprisingly enough. My daughter, yeah, it's my daughter. She runs hot. Now, she knows she runs hot. That means that her temperature is always about 99.5. Right. And her nurse at school has never taken her temperature at a normal level. So whenever she wants to come home from school, she goes to the nurse. And the nurse takes her temperature and like, oh, she's running a fever. You have to come pick her up. Yeah. Like, stop it. You're giving in. She's playing you. They're like, I'm sorry. Rules are rules. She's running a fever. Nah. For her, or for Reagan, if your temperature runs at 99.5, your enzymes are evolved to work best at that temperature. Some people, 98.1. Some people, 97.4. Yeah, 96. Some people run low. Your enzymes are evolved to work absolutely ideally at those conditions. Here, we've got this is a graph of, this actually looks like human body temperature here. 37 degrees Celsius is human body temperature. Starting off really, really low, you'll notice there are low reaction rates. These molecules cannot connect with each other. As you increase the kinetic energy, the molecules begin to interact. They begin to cause reactions. That steadily builds until you reach what's known as the optimum temperature. The reaction rates are the fastest they will possibly be. Once you get beyond that optimum, the rates drop dramatically because you start to lose the shape of the enzyme, the conformation of the enzyme. So at low temperatures, you have low kinetic energy. The molecules and the enzymes aren't meeting. As you increase kinetic energy, you start to have the enzymes meeting the substrates, and you have reactions occur. Faster and faster until you reach that optimum temperature, at which point you get a sharp decline in the reaction rate. At very, very high temperatures, the bonds holding these proteins together break down. What are 5 forces that hold proteins together? Covalent bonds. Van der Waals forces.
Yes.
I figured that's what you're saying. What? Ionic bonds. Covalent bonds. We got that. Hydrogen bonds. And the hydrophobic effect was the 5th one. At high temperatures, those bonds start to fall apart. So we go from having a 3-dimensionally folded up protein into what's called a denatured protein, a protein without those bonds. It lost the shape. That denaturation cannot be fixed without what are termed chaperone proteins, and that doesn't happen often. So you go from a protein that has this very specific active site. That active site has a certain shape to it. As you increase temperature, the bonds break, and you lose the shape of the active site. So you don't have reactions occurring. Egg. Have you guys fried an egg before? Yeah. Okay, you fry an egg. You crack open an egg. What does the egg look like when you crack it open? It's sort of like clearish, right? And it's got a yolk in it, right? You put it on a frying pan. You increase the temperatures. The egg white begins to denature, and it starts to solidify and turn white. Can you unfry an egg? No, because you can't renature proteins. You lost the 3-dimensional shape. Another condition will denature enzymes, and that's pH. pH, again, is the measure of hydrogen ions. So if you have too many hydrogen ions, those are going to end up pushing against the bonds in the protein until the bonds break down and you denature. If you have too few, you have too many hydroxide ions that will break the bonds in that protein. You have to have just a very, very stable amount. How do you stabilize the number of hydrogen ions in solution? Buffers. I always think, do you guys remember playing Red Rover, Red Rover? Yeah. So with Red Rover, what happens is you form a chain, like an amino acid chain. You're going to have some people running at that chain. They might represent the hydrogen ions. Now, if you had one person running at you, usually you can hold it together. But can you imagine having, like, I don't know, thousands of people charging this chain of people playing Red Rover, Red Rover? They're going to let go and break. Yeah, because you don't want that. So that's denaturation. Well, I wanted to, but it looks we're running late. So those are the 4 big things that will impact enzyme reaction rates. Concentration of enzymes. The more enzymes you have, the faster the reaction rate. The concentration of inhibitors. The more inhibitors you have, the lower the reaction rate. The temperature. An optimal temperature will have the highest reaction rate, while beyond the optimal, reaction rates drop off dramatically. And then the pH, again, optimal pH and beyond optimal pH, reaction rates drop off. One thing I do want you to understand is that at high temperatures, you cannot refix an enzyme. Reaction rates are low, and they will stay low forever unless you add more enzymes. At a low temperature, you didn't destroy the enzyme. They're just not moving fast, which means that if you heated it up, the reaction would begin again. That's going to come into play in your lab. And then finally, a quick slide on cofactors and coenzymes. You've probably heard of cofactors and coenzymes in commercials. Basically, what you have with a coenzyme is that coenzyme is a piece of a puzzle. You have an apoenzyme, part of an enzyme, that will bind to a coenzyme, something that you're going to eat, take into your system or you build, and that will allow you to then fully bind to a substrate efficiently. So coenzymes are going to speed up reaction rates because substrates don't bind to enzymes as well without them. All right, so we've gone into how enzymes work. We've gone into how to speed up reactions. Now we need to get into how those reactions happen, where they happen, and what's going on in the cell. So we're going to talk about cellular metabolism. All of the reactions building things up and breaking them down. Cellular metabolism is the combination of all of the cellular processes across the board happening in the cell. Here is a basic cellular pathway of the breakdown of glucose. You're going, let's see if I can figure this out. Let's see, So right here we have glycolysis. So we're going to start breaking down glucose here. It's going to produce these byproducts. Those are going to be broken down. You'll notice that each of these steps has a little blue line attached, moving from one thing to another. So here you have a reactant that becomes a product. It's a process. Each of those blue lines represents an enzymatic reaction. Specific enzymes are involved. And they're very different enzymes for each. It's almost like a flow chart. You guys ever worked with flow charts before? One leads to the next, to the next, to the next. That's what's happening here. A reactant leads to a product. Now that product is the reactant in another reaction. Then that product is a reactant in another reaction. On and on and on. So what we're looking at is now all of these reactions occurring in the cell. And this looks complicated. The thing is, a scientist could take their entire career just to figure out this one small section. And then call their life well worth it. That is, it seems like a small piece, but there's a lot of work that goes into figuring out each of those little steps. So as scientists work together, they build a more complete picture. Now this is one reaction happening in a cell, one group of reactions happening in a cell. This is not all of the reactions happening in the cell. That last slide was just this little piece right here.
That's crazy.
Pardon? That's crazy? And so we're mapping out the slide, and that it gets even crazier because this is just 1 out of 47 pages that looks like this to map out all of the reactions happening in the simplest cell. Not even in your cells. So it gets complicated very, very quickly. But in the end, it's all just very simple pathways following the flowcharts. This is the breakdown of lactose. You guys have heard of lactose before, right? Where have you heard of lactose? You hear of lactose intolerance. People can't drink milk. Right. Thanks. Lactose is broken down by what's called an enzyme called beta-galactosidase. So that lactose, using an enzyme, and is broken down into galactose and glucose. That glucose can bind with ATP. And when it does, it gets an energetic phosphate attached to it, so it becomes glucose 1P, 1-phosphate. That enters into a process called glycolysis that then breaks down that glucose. Going in the other direction, we have galactose gets broken and gets phosphate added to it to become galactose 1P. That then becomes UTP galactose to become UDP glucose to become glucose 1P, and then it enters in. It's just step by step following the flow chart. Can you skip steps? No, not really. If you skip steps, bad things happen. You can follow in a different route, but if you skip the steps, then galactose 1P cannot become UDP glucose, which means you don't get glucose 1P. I will not ask you to memorize a pathway. What I'm asking you to understand is the idea behind it. On the next test, you're going to memorize this pathway in much, much greater detail. For the next test, not the one coming up, the one on cellular respiration, we're going to cover it next week. In fact, it's the, I believe, yeah, it's the next lecture.
So after this lecture, can we stop setting the exam?
Yeah, this is the, it's a test. We don't want to freak Reagan out. But yeah, this is the last test that will be on this coming, I'm sorry, this is the last lecture that will be on the coming lecture.
Lecture on Wednesday next week after the test. No
I don't know. I'll have to check. So we're going to talk about two types of reactions, and you know them, anabolic reactions and catabolic reactions. We're going to start with the catabolic reactions. Basically, give an example of a catabolic reaction. You destroy things because they're, right, cats. Cats destroy stuff. One example. What chemical reaction breaks things down? Hydrolysis. If it was condensation, I would have asked Sarah. All right, so we're going to, the reason we have these catabolic reactions is several-fold. One big reason to have catabolic reactions is for recycling. It's way easier to reuse pre-built components. Remember, all of our macromolecules are made up of these individual subcomponents. What are those subcomponents called? What are macromolecules in general made of? Monomers. Monomers come together to form these polymers. Each of those monomers takes energy to build. But if we can recover them, we don't have to invest the energy in rebuilding new ones. If I wanted to build a protein, I could start from scratch and build all of my amino acids. Or I could take another protein that's not doing anything anymore and break it apart and rebuild it. Okay. Legos. You guys have, I assume, had a childhood where you at least played with Legos once in your life. If you played with Legos, you build something. You might follow the directions and build that thing that came with the box. What do you do after that? You do your own thing. Well, you can, but the thing is, you don't want to, you've got this one thing you've already built. You're not playing with it anymore, so you can take it apart and build something new. You don't have to invest resources in building those the new subunits. Now, what would happen if you really, really liked whatever you built at first? What would you have to do? You'd have to buy new ones. You'd have to invest energy. Put it on, you're right, put it on a shelf, set it aside, but then you have to go buy new ones. You have to invest energy. You have to invest resources. So it's easier just to break down something that's pre-existing that you're not using anymore to build new components. So that's really what these catabolic reactions are used for, recycling macromolecules. So what is, we've got a bunch of macromolecules, right? What are the monomers of the nucleic acid? Nucleotide. What are the monomers of the proteins? Amino acids. What's the monomer of the polysaccharides? Monosaccharides. Well done. By breaking down these larger molecules, you can get the components to rebuild new large molecules. The other thing, it's not just about recycling, it's about recovering stored energy. When you build bonds, you store energy. When you break bonds, you release it. So they're going to be, if you're releasing energy, it's an exergonic reaction. The energy produced from breaking down those molecules then can get stored in what are termed energy intermediaries, little batteries. Now, we've already talked about one energy intermediary. What was that major energy intermediary that we talked about? We talked about one energy intermediary, one cellular battery, one molecule that we build. Yeah, we have to recharge it, but what is that molecule? I know. Adenosine triphosphate, ATP. Well done. ATP. So it's the next slide, I know, right?
That rhymes. ATP, the rechargeable battery.
Haha, they can remember it. So ATP can be hydrolyzed. You're going to remove a phosphate, releasing energy. That's a catabolic reaction. Or it can store energy by being built. It can store energy as an endergonic reaction, a condensation reaction, to go from adenosine diphosphate to adenosine triphosphate. There are two broad mechanisms for building adenosine triphosphate: substrate level phosphorylation and chemiosmosis. Substrate level phosphorylation is where you use an enzyme to directly build adenosine triphosphate. You take an adenosine diphosphate and a phosphate, and you squish them together. Diphosphate and a phosphate, you squish them together, now you have triphosphate. So that's it. Substrate level phosphorylation is where you use an enzyme to build ATP. That is the whole definition. So when I inevitably ask you, what's the difference between substrate level phosphorylation and chemiosmosis, part of that answer is substrate level phosphorylation requires an enzyme. All right? Substrate level phosphorylation and chemiosmosis. So the way I remember that is we see substrate. When you see substrate, automatically think enzymes. Enzymes go with substrates. And phosphorylation means adding a phosphate. So that's the easier one. Substrate level phosphorylation requires an enzyme to build ATP. The other mechanism for producing ATP is called chemiosmosis. Chemiosmosis is where you're going to use the energy stored in an electrochemical gradient to make ATP. Okay, now we've got to remember what a gradient is. So what's a gradient?
The way the
Solute?
yes.
Solute will follow its concentration gradient. So if you have a high level of solute in this space here, it's going to want to get into that space there. It wants to go from high to low concentration. It wants to diffuse. Make sense? So what we do in our mitochondria, aerobic cellular respiration, how we produce ATP is we're going to create a very, very high gradient in that space, in that intermembrane space. Because remember, mitochondria are double membrane bound, an inner membrane and an outer membrane. So between the two membranes, you have a lot of hydrogen ions, tons of them. Those hydrogen ions want to get into that matrix. They want to get on the inside. The only way they can get through is through that molecule there called ATP synthase. ATP synthase is a channel. It will let these hydrogen ions through. But as they go through, they spin it. They make it rotate. The energy, the kinetic energy of that rotation is then translated or transferred to take an adenosine diphosphate and a phosphate and squish them together. So the idea is, you guys know what a water wheel is? Okay, a water wheel as it spins, you've got river water running like, river water's going this way, so the water wheel is spinning like that. Okay? That kinetic energy can do something in a wheelhouse, whether it's hydroelectric power or whatever. What the ATP synthase is doing is it's spinning just like that water wheel, and it's turning that energy into the bonds of ATP. That's the simple explanation. We're going to get into this in a lot more detail in the lecture on cellular respiration. It may make more sense then. I just wanted to introduce you to the concept because this plays a major role in both photosynthesis and aerobic cellular respiration.
So we're going to have to know everything you told us.
You need to know that. Yeah, no. Simple definition for this particular test. For the next test, I will expect you to explain it in detail, probably as an essay.
What's the essay you use this test?
I haven't written it. No essay? Let's see, what have we talked about? Oh,
Word bank.
well, we'll talk.
Word bank. Actually, you said that one of these is or the cell.
Oh, well, that's going to be the way a protein is embedded in the membrane. There's probably going to be a short answer for 3 points.
What about like the cell theory?
Cell theory, 3 points. Yeah, I'll figure it out. I'll have it written by next time we meet. Okay, so we need to move energy from one place to another. We have to create this adenosine triphosphate. You have to understand with energy, what we're dealing with ultimately is the movement of electrons. What happens when an atom absorbs energy? Not really, but
Electron
The electron cloud expands because those electrons are moving faster. Now, if you took those energetic electrons, those electrons that are moving really fast, you can move them from one molecule to another. And then they could move from this molecule where they were doing something, take that energy with them to another molecule and do something new. Just like when you take the remotes, the batteries out of your remote to power something else, your remote no longer has power, but you have something else that can do work. So this moving electrons, if you were going to lose an electron, an electron is lost, that's called oxidation. If you receive an electron, that you take that electron in, that's called reduction. And there's some good reasons for that. Because it really, on its surface, doesn't make much sense when you're adding an electron, you're gaining an electron, you're reducing. But the thing is, think about the charges. If you add an electron, what charge does an electron have? Negative. So if you add a negative charge, what happens to the charge?
More negative.
Yes, the charge goes down. The charge is reduced. That's where that word comes from. With oxidation, oxidation is all about losing electrons. All right, what do you guys know about oxygen? We breathe it. It has a very high what?
Polar.
That's water. But it's the property of oxygen that makes water polar. Has a high what? Starts with an E. High electronegativity. So it pulls electrons to itself, right? So if oxygen's coming by this sodium here, it will steal electrons. So that sodium got oxidized. Yes, this is stupid, but check this out. So it's like, let's say this is an electron, right? It's hanging out by you. You're sodium, I'm oxygen. I come through and I'm like, oh, you've been oxidized. The way I remember is Leo goes grr. Leo being lion, right? So lose electrons, oxidation. Leo, lose electrons, oxidation. Gain electrons, reduction. Grr. Because I have to gauge where the class is based on where you are. I figure out, like, I look around. So in case you guys didn't know, when I look at different people, I'm gauging where the class is. So you're one of the people I look at. I look in the back. I check Gabby. If you're on your phone, then I've lost everyone. It's like, that's why I look at different people. No problem. You're one of the ones I look at. I check out Sarah, too. She's right there. Annie tends to know it, so I don't have to worry about her. Oh, no. I am really proud of you. Like, you're blowing this thing away. Lose electrons, oxidation. Gain electrons, reduction. Now, if you've been in chemistry, you may have heard of another one. I don't care how you remember it. You just have to remember it. Chemistry, they like to say, like, oil rig, because it's oxidation is loss, reduction is gain. Yeah.
So wait, the G.
Which one? Leo goes grr. Leo. So lose electrons, oxidation. And then grr is gain electrons, reduction. Leo goes grr. Gain electrons, reduction. And moving energy through a system is all about the oxidation reduction reactions. This is oftentimes shortened to say redox reactions. So because oxidation reactions will always go with reduction reactions. One doesn't happen without the other. They're combined. They connect. Where one atom loses electrons, another necessarily gains them. We have a bunch of examples in nature. One is rust. If you were to leave iron outside, what happens to it? It turns to rust. It goes from being one particular chemical state to another, because oxygen's coming along and stealing electrons. In the Krebs cycle, the Calvin-Benson cycle, which we'll get to in photosynthesis, an entire stage is devoted to reduction. But then we have, this is one of my favorites, the Statue of Liberty. What's the Statue of Liberty made of? What color is copper? What color is the Statue of Liberty? Why? Oxidation.
Oh, so originally it was?
Yeah.
Yeah, it was brown at first.
It was originally brown. What happened? We stuck it out in the middle of a bay with a whole bunch of oxygen and water around it. What happens to a penny if you let it sit too long? It turns green. Same reason. Because what's happening is the copper is getting oxidized. So it all comes back to who cares? This is chemistry, right? The reason that we care about this in biology is because oxidation reduction reactions can be used to produce energy intermediaries, like ATP. So it's all about moving that energetic electron. A new energy intermediary I want to talk to you about is NADH. So NADH, just like ATP, is produced through exergonic reactions. And it's used to donate electrons, donate energetic electrons to other reactions. Makes them happen. So if you think of ATP and NADH as the energy currency of the cell, NADH is a quarter. ATP is $1. ATP has more energy associated with it and can do more things. But there's some times where you just need a little bit of energy. So you don't want to waste that whole ATP. You don't want to break $1. Nobody wants change. Does that make sense? One of them has higher energy than the other. Both do roughly the same thing. Both power reactions. Both move energy from one point to another. It's just ATP has a higher level of energy than NADH. So that is catabolic reactions, breaking things down. Now we'll talk about anabolic reactions, building things up. So whereas catabolic reactions are going to break down and release energy, anabolic reactions are going to require energy. So where catabolic reactions are exergonic, anabolic reactions tend to be endergonic. Now I'm not going to go into much detail about this because really it's just catabolic reactions backwards. So if you understand how oxidation reduction works, it works the same way with this. That's all I have to say about that. Catabolic and okay, so if you took all of the catabolic reactions and all of the anabolic reactions, you put them together, what do you get? Your metabolism, right. What is an example of an endergonic reaction? Hey, Sarah. A condensation reaction. What's an example of a catabolic reaction then? Not condensation. Hydrolysis. All right, so now we have all these reactions happening, but we can't have them happen all at once all the time. We need to regulate them. We need to make sure the right reaction turns on at the right time. And there are three ways, threes, right? 3 ways of regulating a metabolic pathway. Genetic regulation, cellular regulation, biochemical regulation. Genetic regulation is manipulating the DNA directly. DNA produces RNA. The RNA goes on to produce a polypeptide. If you can turn the DNA off, you've stopped the whole reaction from happening. By turning off the DNA directly, you stop the entire metabolic pathway that it runs. If I wanted to turn off the lights in this room, there are a few ways I could do it, all of which are equally effective. What's one way? That's true. You can unscrew them. I could turn the light switch off. I appreciate your creative thought, but that would have been my light switch. Another one is I could turn off the power to the whole building. I could just shut the whole thing off. That would be like manipulating the DNA, turning off the master switch. Or I could break things, I guess. You know, that's cool. Unscrew them, which is less breaking because then I could reuse them. Like the back to batteries. So genetic regulation is directly interacting with the DNA. Cellular regulation has to deal with the membrane. It's all about, if cellular regulation is being regulated by a signal coming in from the outside. A pathway won't turn on until you get a signal from outside of the cell. And that signal from outside then interacts with a pathway on the inside. It starts a process. Cellular regulation is also compartmentalization. Where do you build most of your ATP? What organelle is responsible for that? The mitochondrion. It's compartmentalized. That's where it happens. It's a specialized area. And you give it a very special environment so it happens. How about your hydrolysis reactions? What are most of your hydrolysis reactions? Or your, where are things broken down in the cell? Nope. These things, they happen in the cytosol, but what's like the recycling organelle? The lysosome. So the lysosome is going to have a very specialized environment. It's got all of these hydrogen ions in there and these acid hydrolases. The rest of the cell doesn't have that.
Clean it up.
Right, cleans up the cell. So that's compartmentalization. Those things all happen in one area. That's called cellular regulation. And then you have biochemical regulation. With biochemical regulation, this can be run through either feedback inhibition or controlling a rate-limiting step. I like feedback. Feedback inhibition is the idea that a product later on in a pathway can interact with an earlier step so that that product is no longer being made.
Well, which one?
So if you have that orange to purple to blue to red, that red will interact and act as an inhibitor, say, to the enzyme between orange and purple. So now that orange is no longer becoming purple, so you don't have much more red made. So if you have high concentrations of red, you don't need any more red, so it stops the process. Okay, so there's a series on like the Learning Channel where there's a family that had like 1,001 kids. Do you guys know what I'm talking about? Not like 1,000. It was like 20 something. Yeah, but I forgot the. Yeah.
Oh, yeah,
There was like 21 and counting, something like that. They had a ton of kids. Right. They stopped counting because there were too many kids. Okay, so here's the way it works. You have the mom. She produces a kid, a baby, who becomes a child, who becomes an adult. Right? They're still producing a bunch of kids while that one is going through these different stages. Now, this adult decides they don't want to have any more brothers and sisters. So they go back and they slip their mom some birth control. Right? So that stops the process. Does that make sense? Like, here, try these Tic Tacs. It stops the whole process so that doesn't kill all the kids that are in already being through the system, right? They still grow up to become adults, but now we've stopped the process back at the beginning. That's called negative feedback. Positive feedback is going to go in the other direction, though. You produce a final product that interacts positively with the beginning. It speeds up the reaction. It might be a catalyst. If that's the case, you have same idea, mom, kid, teen, adult. The adult figures out a way to convince their mom to get fertility treatment. Well, now she's churning out even more kids. It's going even faster. So you have positive and negative feedback, where one step is interacting with another. It all comes down to regulating these steps. The only step that matters that you regulate is what's termed the rate-limiting step. There is one step that will be the one that sort of controls the pace of the whole thing. By controlling the speed of the rate-limiting step, you control the overall reaction. If you were to make burgers, What is the rate-limiting step of making burgers? What is the slowest part of making burgers? Grilling the patty, making the meat, right? It's really easy to throw out a bunch of buns, just chuck them on the counter. Add, take the mayonnaise and mustard and just go like that. You can do that really quickly, but each of those burgers takes a finite and defined amount of time to cook. If you were able to speed up how fast you put out buns, would that speed up how many burgers you could make? No. What matters is if you could speed up how fast you cook the burgers. They are the rate-limiting step. If I made you stay in lab until all of the lab groups were done, it doesn't, I know, don't groan about it. The one that was slowest would be the rate-limiting step. It doesn't matter how fast the other ones get It doesn't matter how fast all the other groups get done. What matters is how fast the last one gets done. So if you speed them up, you speed up the whole reaction. Okay, so we got to recycle these things. Most molecules, except for DNA, get recycled really quickly because you don't want them to build up. Take like mRNA. mRNA's whole job in life is to take a plan from the nucleus to the ribosome. After that, you don't want to keep it around. It's like hoarding things. You guys seen the show on hoarders? Usually it's like a crazy old cat lady, or they've stored all these newspapers. What happens to their house? It becomes a maze. They run out of room in there. It's all filled. The cell can't abide that. You cannot have that happening because the cell will eventually break down without space. And you've got so much energy tied up in those macromolecules, so you have to recycle them. So, here's another quote from you guys who were good at that Gadsby quote. What do you do with a general when he stops being a general? Where is that from? Do you know? It's not from a commercial. No, it's actually from White Christmas. Bing Crosby. No, have you guys seen White Christmas? Do you know what I'm talking about? No. Okay, then never mind. So it's different generation. What? Where? Nice. I appreciate how you set up for that too. All right, so you got to get rid of this RNA. RNA is what kind of macromolecule? What kind of macromolecule? There are 4 kinds of macromolecules. What are they? Proteins are 1. What are the other 3? Carbohydrates, another. Lipids, 3. One more. We got protein already. Nucleic acid. So what is the monomer of the nucleic acid? The nucleotide. Well done. She's good, yeah. So that mRNA is broken down by an enzyme called an exonuclease. It's going to cut one nucleotide off and then another and another and another. One at a time. It speeds up hydrolysis reactions. Exonuclease. Exo, so you're cutting it off. Cutting it out. Yeah, of nucleotides. So it just cuts off these nucleotides one at a time. Cool thing is that doesn't happen fast enough. So what the cell does is it builds a complex of a whole bunch of exonuclease. So if one was cutting things fairly efficiently, you get a bunch of them together and you build what's called an exosome. And an exosome will utterly devour a nucleotide. It sort of starts on one side and just chomps its way down. So that cuts up mRNA and releases a whole bunch of nucleotides. RNA goes in one side, nucleotides come out the back, and that lets you build more RNA with less effort. So that gets rid of RNA. Proteins are another thing that builds up in high supply. So we got to get rid of those. In order to cut up a protein, you're going to use what's called a protease. Please do not call it a protease. It's spelled that way, I know. It's a protease. So you cut the peptide bonds between amino acids. And if you get a whole bunch of proteases together, you get what's called a proteasome. And a proteasome will start on one side of a polypeptide and start spinning its way like a buzzsaw down it, hacking off amino acids. So you got exosomes and exonucleases that break down nucleotides, proteasomes and proteases that break down proteins. The question becomes then, how do you figure out which is ready to get rid of and which one is a good one? And that's where ubiquitin comes into play. Ubiquitin is a little red flag. It's not a literal red flag. It's a chemical signal. And what that signal does is it starts to attach to nucleic acids and proteins. So it sort of like starts binding on one end. Comes along, binds. As you build up more and more and more of these ubiquitin molecules, it becomes a bigger and bigger flag. It's sort of like the older you get, the sadder you become. How's your life? Is that what you said? The older you get, the sadder you become. So when you find somebody who's really, really, really sad, it means they're really, really old and ready to go. Or they're ugly. So the older you get, the more wrinkled you become. You get more wrinkles. Thus, you know somebody's really old when they have a lot of wrinkles. And they're really sad. You guys are seriously depressed. So if you've got a whole bunch of these ubiquitin molecules building up, you know that it's time to break that molecule down. Now this ubiquitin works because it automatically attaches to improperly folded proteins, so things that can't do their jobs. And if you need to remove proteins quickly after like the cell cycle, which we'll get into later, ubiquitin binds and lets you tag these proteins for removal. So once that ubiquitin binds, an exosome or a proteasome will bind to the ubiquitin and devour the molecule. Sometimes you have to get rid of a whole organelle. You have to recycle a whole organelle. A mitochondria gone rogue. Because mitochondria are going to independently reproduce. So if you've got a bad one, you want to get rid of it. Otherwise, it's going to produce a whole bunch of bad ones. So what you do is you form a vesicle around that bad organelle, and then you bind a lysosome to it. The lysosome does its work, breaking it down. If you don't get rid of bad organelles, there are consequences. They might go on to reproduce. That's how we get Huntington's disease. Huntington's disease is caused by the cell not being able to get rid of broken mitochondria. Those broken mitochondria reproduce, and they reproduce, and they reproduce. And because of that, that's why with Huntington's disease you have all sorts of issues with energy. Lots of shaking. Energy in the cells is way off. So you have to get rid of broken organelles in order for the body to continue working, or the cell to continue working effectively and functionally. Whew, that's a lot, right? Any questions?
Yes.
What are your questions?
No, I'm excited.
You're excited because it's over? DNA does not. So the question is, does DNA break down by exonuclease? Usually not. DNA is broken down using. You do use exonuclease to cut out little pieces of it, but usually not the whole strand. Mainly because it's so stable.